This is the first part of a series of webinars that are going to be covering the blood. The first part will cover or be a general overview of the components of blood and their functions, and spending more time on red blood cells. Subsequent webinars will cover blood typing, hemostasis, and the other components or cellular components of blood. So what we're going to do first is kind of start with a little general overview. Remember in AMP1 we covered connective tissues, and blood was considered a connective tissue. It's a fluid connective tissue, and the fluid component of blood is referred to as plasma. So I'll write that down for you. Plasma. Now remember, the reason why blood is considered a connective tissue, connective tissues, you have cells, but you also have these protein components or protein matrix. You're like, how does blood have a protein matrix? It has fibers in blood that are normally soluble under normal conditions. So we're going to be looking at that specific component that becomes insoluble under certain conditions. Now, blood has a variety of functions. It transports hormones and nutrients and gases and a number of different regulatory components. It's involved in homeostasis. It will be important in body temperature regulation, the maintaining composition of different components, say in your interstitial fluids. It also will be important in protection, but protection kind of falls under the guise of homeostasis. So it's going to be important in a process called hemostasis, which helps you restrict blood loss, or you would most think of blood clotting. It also is going to be, because of some of the components in blood, will be important protecting us against microbes and viruses and various toxins. The study of blood, or individuals who specifically will study blood and its disorders and the different components of it, are referred to as they're hematologists, but the study is hematology. Okay, so those are the people. This refers to as when you talk about blood. So let's just first look at kind of some of the characteristics. As a general rule, I mean, it's about 7% of your body weight, but most people just think about blood volume is about 5 liters, give or take. So it's pretty close to, when we look at average cardiac outputs, average cardiac outputs is 5 liters per minute. You pretty much pump your entire blood volume around in your body within 1 minute. It's pretty cool. Men have greater blood volume, gives them an advantage because they've got more blood. The general characteristics, it's generally about body temperature, core body temperature. And we learned in AMP1 about body temperature regulation. If blood temperature increases or decreases, there's certain mechanisms that take place to try to get body temperature back into the normal range. The blood itself is slightly more viscous than water, and the reason why it's slightly more viscous is because of the formed elements, the different cellular components in the blood. And remember, in discussions of the cardiovascular system, viscosity can affect blood flow. So if blood is too viscous, it can decrease blood flow. And a thing that would cause a change in viscosity would be changing the number of the blood cells. Also, blood has the average pH of blood is 7.4, but you'll see that I put a range because it depends on where you're sampling the blood. The 7.45, you'll see this in arterial blood if you sample arterial blood. If you sample venous blood, it's about 7.35. The reason for the lower pH in venous blood is because of its carrying carbon dioxide. It affects the pH, it lowers the pH. So what we're going to do first is I want to talk about some of the clinical uses of blood, or the analytical uses for blood. Blood is a common thing that's taken to evaluate the things like their electrolyte levels. Do they have too much or too little red blood cells? It's a very common thing to do. And phlebotomy is the term for when people collect blood for sampling. A phlebotomist is the person that will draw a patient. And there's different types of draws that they will do. And so I want to discuss a few of them. And so first I'm going to do something called a venipuncture. So a venipuncture is very commonly used. Most of you have probably had this done. If you went in to have some blood samples just for general workup, is most likely they took a sample from a vein. And the most common vein that they sample from is the median cubital vein. So they could see it right here, the median cubital vein. It is other veins that there's generally like 3 veins that the phlebotomist can draw from or more likely to draw from: median cubital, the basilic vein, and the cephalic vein. But this is the one that they most likely want to do. So where in your notes it says advantages, I'm really referring to the advantage of drawing from the median cubital vein. This one is the easiest to locate on most people. It's not close to any major nerves or arteries. It's less painful, and it's well anchored, so they're not going to be digging around trying to get to it. If you look at the basilic vein, they don't like to have to draw from the basilic vein because it's a little close to the median nerve and the brachial artery, and it's very difficult to anchor it, so it's going to be a little more uncomfortable for the patient. Now the cephalic vein, though, is something that if it may be the actually only vein that's palpable in a person that's obese, so they may have to go to the cephalic vein. But again, they don't like to go for it because the median cubital vein is usually the easiest to get. It's the less painful or uncomfortable for a patient. Now you can get, when you do a venipuncture, you can get a fairly amount, like was a lot of times, like last time I had a blood drawn, they took like 5 tubes of blood. You can get a fair amount of blood, and they may do that because they're going to do a number of different tests on you. But if they only need a very small amount of blood, they can do something called a heel stick or a finger stick. The heel stick is commonly done on newborns for newborn testing. The heel stick and finger sticks are very minimally invasive, and doesn't hurt that much, but it only works if you can utilize capillary blood. So whatever you're measuring must be able to use capillary blood, not venous blood. So it's capillary blood. Commonly, a lot of people do finger sticks for monitoring blood glucose levels. A lot of the tests for inborn errors or genetic testing for children or inborn errors of metabolism, they do the heel stick. But nowadays things have become really, some of the new techniques and tests have allowed them to utilize a very, very minimal amount of blood, and by using the finger stick or heel stick. So a lot of blood tests can be used with just a heel stick or well, in adults we don't do heel sticks, but finger stick. Now arterial puncture or arterial stick, here you have to sample it from an artery. Now these arteries are much more uncomfortable. The vessels are deeper, they're thicker, they have more nerves associated with it, but sometimes an arterial blood sample must be taken because the makeup of blood does change when you go from, say, arterial to venous blood, primarily in the amount of dissolved gases. So if they want to test blood before any of its contents have been used by body tissues, they're going to do an arterial stick. So sometimes they will do an arterial stick too. So if you look at this picture here, kind of is a big overview of looking at the composition of blood. If you look at a sample, majority of it is the liquid component, plasma. Then we have the formed elements. The formed elements are the cellular components. So we got platelets, you got white blood cells, you got red blood cells. So we're going to look at a couple things first, and then we'll look at the We're really not going to look at the white blood cells in this webinar. We'll look at them in a subsequent one. We'll look at the red blood cells a little bit later. But I want to do first is, so here's like, they're doing a sample. They centrifuge it so you can see how it separates out. You got the liquid at the top here. The buffy coat is the white blood cells and the platelets, and the bottom is red blood cells. So you kind of notice that majority of the cells in blood are red blood cells. And if they want to test how much of the blood is formed elements, they were doing a hematocrit. Hematocrit is also often abbreviated HCT, which actually you see it right here, or they call it packed cell volume. The hematocrit is a percentage. It's important to realize it's a percentage of your blood that contains the formed elements. Now, since most of the formed elements are red blood cells, often when they're looking at hematocrit, they're trying to evaluate if someone has way too many or too little blood cells, but other things can affect the hematocrit. But it's a percentage of your blood that is formed elements. So one thing that can cause, say, a low hematocrit. So you see here someone who's anemic, has anemia. Anemia, it would be they have too few red blood cells. So that would be, okay, that makes sense. Lower percentage of formed elements. Most of the formed elements are red blood cells. So that can cause a low hematocrit. So a low hematocrit is the percentage of the cells in the blood is below the lower normal limits. And it's usually referred to as the red blood cell percentage. High hematocrits, this is when the percentage of the blood cells are higher than normal, and they often refer to as the red blood cells. High hematocrit can be seen in smokers or in people with called polycythemia. Polycythemia is where you have way too many red blood cells. Smokers can have higher than normal red blood cells, because we'll be looking at subsequently because of the effects of, say, carbon monoxide and oxygen transport. But also, high hematocrits can be seen in dehydration. So this is why I want to go over this one, because it illustrates that hematocrit is a percentage of that blood that is formed element. So I'm going to just draw a picture next to the normal one. So here's our tube. And this, you'll notice I'm drawing in, they've got the same blood cells, number of blood cells. But what I'm going to do is I'm going to lower this to say here. So the percentage, so here's the whole thing, the percentage of blood cells in that sample are higher because we've lost some of the fluid component in people with dehydration. So they have high hematocrits. So either can cause issues. High hematocrits can affect increased blood viscosity, which could affect blood flow. Low hematocrits, if you don't have enough blood cells, your oxygen carrying capacity is going to be affected. Now, if we look at this picture, I'm going to look at what's in the plasma, the liquid component of blood. Now, a lot of this is in your notes. I'll just cover that first, and we'll go over the ones that you need to fill in. But you'll know plasma has your electrolytes, like your sodium and your potassium and calcium. Got your water. I do want to mention the water. Water is an important component because the characteristics of water make it perfect in temperature regulation, its role in metabolic reactions. So water is going to be a huge component of that plasma. Blood's carrying, or the plasma carries waste products. You got to take it to wherever you need to get rid of. Got all your nutrients and your vitamins, your hormones, and you got your gases. Okay, so those we got. Let's look at those proteins. So the proteins, there are 3 major proteins we're going to look at. All of them are produced in the liver. Albumins, or albumin, is the majority of them. Albumin, one of the things it does is it maintains osmotic pressure. You should have learned that in AMP1. If you had me, you had it. You learned about osmotic pressure. It also will serve as a transporter, transports things like hormones, which we covered during endocrine system, and things like, say, and I'm going to abbreviate it, fatty acids, FA, so fatty acids. The globulins, you have gamma globulins, that's the Greek letter gamma. Gamma globulins are your antibodies, and antibodies are going to play a role in immunity, in your immune system. So we'll be looking at that in the next lecture. The alpha and the beta globulins, these are transport proteins. So you see we do a lot of these proteins are from just transporting things throughout the bloodstream. Now the last one is fibrinogen. Fibrinogen is a soluble protein. This is that protein, part of that protein matrix that you have in blood that makes it the connective tissue, but this is converted to an insoluble component called fibrin during blood clotting. So fibrinogen plays a role in blood clotting or hemostasis. So fibrinogen, which is soluble, will be converted to fibrin, and we're going to discuss that in a lecture by itself when we do hemostasis. Now the cellular components, I want to just introduce you initially to the cellular components. We're going to spend more time about the red blood cells. We'll look at the other ones at a subsequent webinar. Now, you'll notice majority of the cellular components in blood are red blood cells. The fancy term for a red blood cell is an erythrocyte. Cyte means cell, erythra means red, so it's a red blood cell. A lot of them. You get different ranges. You notice this says number per microliter of blood, and that's a very tiny, 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 tiny amount of blood. Now, the red blood cells are perfectly suited for transport of gases, specifically oxygen and carbon dioxide. Now, the white blood cells, or the leukocytes, so leuko means white, so white cells, these are involved in, we call, actually, let me just write them as nonspecific and specific defense systems. When we do lymphatic and the immune system, this is where we're going to talk about them a lot. The last one are platelets. Now, platelets are actually fragments of cells, and they're going to be important in blood clotting. So, majority of the formed elements in blood are red blood cells. The second most common will be the platelets, and then it's going to be followed by the white blood cells. Now, the scientific name for platelets are thrombocytes. That word, knowing that word will be important later. But thrombocytes, thrombo means, literally means clot. So these are involved in blood clotting. So what we're going to do is we're going to spend some time just talking about the red blood cells. So first, if you look at this slide, you'll notice here, this is a blood smear. If you look really carefully, once in a while you see little fragments, little dots. Those are the platelets. In this particular view, I don't see any white blood cells, but I see tons of red blood cells. So to kind of try to illustrate you there, in a blood smear, most of them are red blood cells. Second most common are the platelets, and then you've got some white blood cells. In this particular smear, we don't see any. If you look at them carefully, you can kind of see they're kind of lightly stained in the center. The reason why is because of its shape. They're biconcave discs. They are kind of flattened in the center. So on the smear, they look like they don't have any color, but it's because it's flattened in the center. They are perfectly shaped for a reason. If you look at this picture here, they kind of, in capillaries, they run in rows. They kind of like stack. There's a term they call it rouleau formation. Is they kind of, in capillaries, will line up one by one, and capillaries are so tiny that they have to be able to deform and squeeze through it because they don't have a lot of room. So one of the advantages of having the shape that they are is it allows them to easily squeeze through capillaries. One of the things we're going to look at later is if something's not quite right with its structure, that it's not going to be able to easily to squeeze through those capillaries and cause some problems. Another reason for the shape, this kind of flattened center, is that it's an adaptation to increase surface area. And the surface area is in regards to diffusion of gases. So it helps adapt to increased surface area for gas diffusion, but also the shape allows it to deform easily or squeeze through capillaries. In the red blood cell, we have something called hemoglobin. Hemoglobin is a structure that is composed of 4 globin chains, which are protein chains. And in an adult, normal adult, we've got 2 alpha and we've got 2 beta chains. In the center of each of these chains, we have a heme ring. And that heme ring, at the center of it, you have iron. So you got 4 protein globin chains and a heme ring, which in the center is iron, which binds to oxygen. So each molecule of hemoglobin can bind up to 4 molecules of oxygen. Now, what would happen if the structure was somewhat not right? Well, we can end up with something. A good example would be sickle cell disease. Sickle cell disease is person, we're going to look at this here, this child has inherited 2 defective genes for hemoglobin. Normal hemoglobin, adult hemoglobin, is called hemoglobin A. This child has inherited 2 abnormal hemoglobin S, and one from his dad, one from his mom. And this defective hemoglobin can result under certain conditions that red blood cells become really hard and sticky, and they end up forming these kind of sickle things. It's a C, kind of like a C-shaped structure that it forms a farm tool called the sickle. That's why they call it sickle. The cells will die early, which causes a shortage of red blood cells, hence the term anemia, which impairs their oxygen carrying capacity. But also because of the shape, they can block blood flow, block capillaries, and cause tissue damage. Individuals with sickle cell trait, which you see here, they tend to have normal lives. They don't have the problems as children with sickle cell disease. They've inherited 1 normal gene of hemoglobin and 1 abnormal. It is a inherited condition, and it's in sickle cell trait. You're a carrier. So if you see here, the mom and dad each were carriers, and they had children, and with that, there's a 1 in 4 chance that one of their children will be born with sickle cell disease. Another thing I want to look at is discuss the lack of organelles in red blood cells. There's a reason. It's not that anything's wrong with them. There's a reason why they don't have organelles. Is the red blood cells need room to carry oxygen. That's their whole goal in life. My job is to carry oxygen. I don't want room, any space taken up by any organelles. So what's the significance, though, of lacking some of the organelles? Well, without centrioles and ribosomes and all that, they don't divide, they don't grow, they can't make any new proteins. So they're going to have a limited lifespan. So you kind of have to pretty consistently replace your red blood cells because they don't live very long because they can't fix themselves. But it's okay. The thing is, they needed the room to be able to carry the oxygen. Their lack of mitochondria they did not want mitochondria because mitochondria uses oxygen for cellular respiration. And they would be taking up the oxygen that these red blood cells need to carry to take to other tissues in your body. So because it lacks mitochondria, it still needs energy, though, needs ATP, so they use anaerobic means, which would be glycolysis. Now here, do not memorize these numbers. I don't have them memorized. Is a red blood cell count. If someone does a red blood cell count, they're trying to evaluate the oxygen carrying capacity of the blood. And the red blood cell count is higher in males than females. It's true. But there's ranges. But if someone's red blood cell count's too high or too low, it can indicate there's a problem. Someone with anemia would have a low red blood cell count. Someone with polycythemia would have a high red blood cell count. Now, polycythemia does not mean it's like, oh, they've got more oxygen carrying capacity. Unfortunately, with polycythemia, is they have high production of red blood cells, but they're not functional, they're not mature. So that leads us into kind of looking at the red blood cell production and turnover. So before we look at its production, let's just look at the turnover. Remember I'd said that they have kind of limited lifespan because they can't fix themselves, they can't repair themselves, they lack those organelles. So the average lifespan of a red blood cell is about 120 days, which is about, what, 4 months or so? And so the red blood cells, we'll talk about where they're produced. You can see, you know, bone marrow, yeah. We'll look at that later. But the blood cells, after kind of they're worn and torn and worn down, is you have macrophage in organs like the spleen and the liver and even bone marrow, because some of the blood cells didn't quite make their way out, so we got to kind of, they weren't quite right, so they got to get rid of them. But the old and damaged red blood cells are going to be broken down. But we're very green. We like to recycle. The hemoglobin is going to be broken down into globin, which you don't see here, but the globin will be then broken down to amino acids, and the amino acids can be used to make hemoglobin again, the globin chains. So the hemoglobin is broken down into globin, but also heme, which you see here. The iron in the hemoglobin is recycled, so it's carried by transferrin, which literally means to ferry or carry the iron, and it's going to be stored in the liver as ferritin, and we'll use it again as needed. The heme ring itself is going to be broken into these pigments called biliverdin, and then eventually into bilirubin. Bilirubin is then transported to the liver, where then it will be, things will be added to it, which will make it a little bit more water soluble, where then it can be transported to other areas in the body, like the kidneys and the small intestine, where then will be kind of subsequently acted upon. And you'll notice, like here in the intestine, the bilirubin is actually broken down into these things called urobilins and stercobilins. Those are actually what gives feces the color that they are. Some of the breakdown products of bilirubin are also removed in the kidneys too. Now let's just go back to this, the red blood cell formation. So you'll notice I have this picture here. It looks like the proximal end of a long bone, but I don't like the fact that they did that, because you're going to see very subsequently why I don't like that they did that. Is erythropoiesis is the formation of red blood cells. In an adult, the majority of your red blood cells are produced in the red bone marrow, but of flat bones, like your ribs, your parts of the flat bones in your skull, your sternum, the scapula. Very little amount in an adult is formed in the long bones, and if it is found in the long bones, it's in the proximal epiphysis. So here's the proximal epiphysis. That's a femur, which you see here. This is the distal, okay, right here. But you'll have it in the proximal. But the majority of the red blood cells are formed in red bone marrow, which is found in spongy bone. So the flat bones have this compact bone, and it has the spongy bone in here. In the long bones, remember the spongy bone is it's at the end here, okay? And then you have the compact bone that lungs along the side. Now in children, or let's say before you were even born, during fetal development, you had blood cells were formed in yolk sac, depending what stage of development you were at. liver, the spleen. You were born. Then you look here. Most of the red blood cells have actually been, in a child, is made in the long bones. So in kids, red blood cell formation is primarily in long bones. In adults, it's the flat bones. But as a child gets older, the red marrow is slowly replaced by yellow marrow. Remember, yellow marrow has a lot of fat in it. Now the yellow marrow gets its color from carotenoids in the fat droplets. This is why they call it yellow marrow. So in the adult, most of the red blood cells are actually formed in the red marrow found in the flat bones. Now, what do we need to make it? What are things that are needed? Well, some important nutrients are needed for normal red blood cell production. Things like vitamin B12, see here, folic acid, and iron. So I'll just give you an example of some things that can happen as a result of not having sufficient amount of these nutrients. Pernicious anemia is where you have a deficiency in the production of red blood cells due to a lack of the vitamin B12. You'll notice the cells, red blood cells, are kind of big. They call them macrocytic. Doesn't mean it's better. So they have insufficient production, and the size is off too. Iron deficiency anemia, very common in females. I had that when I was younger. Is iron deficiency anemia. They don't have sufficient amount of iron. You'll notice that these cells are a little bit lighter in color. They're called hypochromic. They're microcytic. They're a little bit smaller. And some of them are cigar shaped, so they're not a normal shape. So both of them, both conditions, anemia, you have inadequate production of red blood cells, which will impair the subject's oxygen carrying capacity. Now, besides these normal nutrients that are needed for blood cell production, you need certain hormones to produce the red blood cells. And you see this kind of big picture, so I just want to show you this first, and then I'll zoom into what you have in your notes, is all blood cells are derived from hemocytoblasts, which are in the bone marrow. And we're going to follow this coming down here. This is what we're going to do. And what we're going to do, go here, because it's the same thing. So hemocytoblasts, one of the things that can be derived from hemocytoblasts is a myeloid stem cell. Myeloid stem cells will be precursors for a number of the blood cells, one of being the red blood cell. Myeloid, the term myeloid is a word that denotes that something's derived from bone marrow. It's an adjective that pertains to, or it's something that's derived from bone marrow. The red blood cell goes through a number of different stages, and what this little red thing here is trying to represent erythropoietin. That's the abbreviation. You're going to see it in your notes. Erythropoietin is a hormone that is needed for the normal, or it's going to stimulate these cells, these precursor cells, to develop into a red blood cell. But you'll notice that it goes through a number of different steps. So you have all these steps that are taking place in the bone marrow, and at a certain point it enters the circulation, and right before it entered the circulation it ejected its nucleus. Remember, we didn't want that. We want as much room as we can to be able to carry oxygen. The blood cell actually finally matures within the circulation. So this whole process, it starts in the red bone marrow, and ultimately these stem cells will be stimulated to become red blood cells. So you start with a hemocytoblast, go back here. Then under certain conditions, we're going to look at it a later webinar, is you get a myeloid stem cell, and then with erythropoietin, we'll get the eventual production of a red blood cell. Now, the red blood cells, or I should say the erythropoietin that you need for red blood cell production, is actually produced in the liver and the kidney, because these organs have receptors that sense if oxygen levels in the blood are insufficient. And so what they'll do is they'll stimulate the release of erythropoietin, which then goes and stimulates the production of red blood cells in the bone marrow. So it's going to increase the number of red blood cells, which is supposed to increase the oxygen carrying capacity of the blood. Well, it's like, okay, that I definitely would want that. So let's look at some of the things that can result in increased erythropoietin. Smokers. Now, smokers, their oxygen carrying capacity of their blood is insufficient because carbon monoxide, it's a component of cigarette smoke, decreases the oxygen carrying capacity of their blood. So smokers have increased red blood cells, and that is one of the things that you can see. We get higher hematocrits and increase in blood viscosity. High altitude will do the same thing. When someone goes to a higher altitude, the oxygen, the ability to get oxygen from the air is decreased. And so one of the adaptations to going to the higher altitude is to increase red blood cell production. And individuals who live at the higher altitudes, like the Sherpas, up in like Mount Everest, the Himalayans, am I got it right? Himalayan mountains and the Mount Everest, yeah, Himalayas, is they have a natural inborn ability to increase their oxygen content in the blood because they have higher red blood cell numbers. People who are anemic, it's like, okay, my oxygen carrying capacity in my blood is diminished, so that's going to stimulate the production of erythropoietin. Is an attempt to increase oxygen carrying capacity. And individuals who've got problems with gas exchange, so oxygen content of their blood may be diminished, so they're going to try to respond and compensate by increasing the numbers of red blood cells. So this was going to be the last part, the first webinar that covers blood. The second one will cover blood typing.